In this video, we'll be discussing about the muscle tissue, the structure of three different types of muscle tissue and their organization. The muscle tissue is a soft tissue that is specialized for contraction and relaxation to produce voluntary or involuntary movements. We see the embryonic development of muscle tissue is through myogenesis, and we have three different types of muscle tissue. First one is the skeletal muscle, second one is the cardiac muscle, and third one is the smooth muscle. Now let's get to the details and see what are the different types of structures that we can see in these three different muscle tissues. We see we have the skeletal muscle, cardiac muscle, and smooth muscle. If we see the general organization of skeletal muscle, we have structures in the form of epimysium, perimysium, and endomysium, as shown in the diagram. The outermost layer is the epimysium, which is the exterior collagen layer connected to deep fascia, and it separates muscle tissue from secondary tissue. Then we have the perimysium. It surrounds the muscle fiber bundles containing blood vessels and nerve supply to fascicles. And the last layer, that's endomysium. As we can see in the diagram, it surrounds individual muscle cells or fibers. It contains capillaries and the nerve fibers. And we also have specialized cells within it in the form of satellite cells, which are stem cells for muscle tissue, and these cells repair damage to muscles. Now moving towards the microscopic organization of skeletal muscle. We see we have the neuromuscular spindle that has intrafusal fibers. These fibers detect the amount and the rate of change in the muscle length and acts as a sensory element within the muscle. These intrafusal fibers along with neurons aid in proprioception. Second structure is the satellite cell, which are the stem cells, and from these stem cells or satellite cells we get the myoblast and then ultimately the muscle fibers or muscle cells. Then within the skeletal muscle we have the myofibril. These myofibrils are the rod-like structures of muscle tissue made up of myofilaments, as shown in the diagram. Then furthermore we have the sarcomere. These are the smallest functional contractile units of myofibrils, as shown in the diagram. And if we further zoom into the underlying microscopic structures of skeletal muscle, we see we have the Z discs, triad, and myofilaments. The Z discs are the boundaries of a muscle sarcomere. The triad shown in the diagram here are the structures formed by T-tubule with the sarcoplasmic reticulum. And we also have myofilaments like actin, nubilin, titin, and myosin. Now let's discuss the cardiac muscle and smooth muscle in a brief manner from here. The cardiac muscle is composed of cardiocytes, which are small uninucleated cells with the absence of triads. First we see we have the Purkinje fibers that are associated with cardiac muscles. These Purkinje fibers conduct electrical pulses to the ventricular muscles. And if we see the cardiocyte structure, we have the myofibril and intercalated discs. These discs are the transverse and longitudinal components that connect the cardiac muscle cells, thus acting as one single unit. Furthermore, myofibril has sarcomere structure and has substructures like Z-disc, dyad, and myofilaments. And finally, we have the last muscle, that is the smooth muscle, which is non-striated tissue because it has no sarcomeres and therefore no striations. It simply has central nucleus, and we also can see myofilaments in the form of actin, intermediate filaments, and myosin. So this is all about the structural organization of all the three muscles. I hope you like the video. If you like it, give it a thumbs up. You can support my work on Patreon or YouTube and make sure to subscribe this channel. Thanks.